Hello, this is Scott Carpenter, and welcome to another episode of the Sasquatch Awareness Project. Today I'm going to do part 3 of the DNA study in a nutshell. It's been a while. Going to get back into it. Let's get back into the nuts and bolts. So, quick review here. We had started into the haplogroups and where the Sasquatch came from. As far as the haplogroups go, and haplogroups are their lineage, geographically, and it's just like when you do your DNA test. And I won't mention any because I don't have, don't want to get copyright strike. But if you do any of those generic DNA tests, they use the haplogroups to tell you where you're from, and that comes from the mitochondrial side. That comes from your mother's side. And let me see here, I need to let's see, I can get it to play. Slideshow from current slide. There we go. So I'm going to jump right in. There were if we go back to the map here, the red dots are all the locations where the samples were taken from, and the blue squares are where they originate. And you can see there's only a couple that originated in North America and Alaska and Canada, and the rest of them come from Europe, Russia, Asia, Africa. And so we go here, and you can see here the North American haplogroups represented the following Native American tribes, and the haplogroup A6L2C, Inuit and Nikavit peoples, the haplotype C, Yakuts, and I'm not going to pronounce that one, EVENKS, haplotype D, the Aleuts people. And then from the rest of them came from Asia, Africa, Europe, and those are haplotypes H and T, V, H1A. That was the sample 26, flesh and hair. And haplotype South Africa came haplotype L0D2A. So also they got 3 full genomes, and they had 30 samples. They were able to do 30 DNA analysis, nuclear DNA analysis of 30 samples, and these results yielded XX, XY, Y, and null. And this was unusual because this was human mitochondrial DNA, and it should have amplified with the human primers, but it didn't. And that was what part of this study is about. What's not there, what's there. It's all, and you can find out a lot, and I've said that before. It's not what's there, but it's what's missing. It's the negative side. It's what should be there that's not, that really opens this thing up. And the referees that were for the journal Nature that were looking at this excluded all the negative results. And you have to ask yourself why. And I think I know the reason why, and so do you. So here is the DNA samples yielded the 4 types: XX, XY, Y, and null. The X chromosome failing to amplify, also known as a dropout, was considered by Dr. Ketchum the most significant of the findings observed with the STR genotype analysis of the, and I can't pronounce that word. I'm not even going to try, but you can see it right there. And here's the table for this. Is a table taken from the DNA study, and the sample numbers and their results. The amylogenin results, that basically is telling you male or female, X, Y, XX. And so, those markers, 16 of the markers amplified, the rest of them failed. So some of the samples indicated a normal human X chromosome, while others failed to amplify completely. So you had these very odd findings for the nuclear DNA. In other words, what should have been there, given that it was human mitochondrial DNA, was not there. The Y chromosome was amplified across the exons. Exons are part of the DNA that are converted to mature messenger RNA. And those results varied and were obtained with no samples successfully amplifying and sequencing across all 5 exons, except the human controls. So what is weird about this is on the Y chromosome, there should have been some amplification, but there wasn't. The only thing that amplified was the human control. In other words, they put, on purpose, they took, I think Dr. Ketchum used her own DNA for that as a control sample. Say, okay, here's a human, so we're going to run human, and then we're going to see what we get with the Sasquatch, and that's the problem. Here's another thing, that's very important. I'm going to harp on this, is human or non-human hair yielded human mitochondrial DNA, but when they got to the nuclear DNA, which comes from the father, they basically took the nuclear DNA and they did what's called a blast search. I've explained that before. And basically when they did this blast search, none of the nuclear DNA sequences matched any organism in the database. DNA that had human haplotypes, in other words, this DNA had human haplotypes and amplified for that. But when they looked in the DNA and GenBank, it didn't match any known organism. Many sequences failed to amplify with human primers. That was another a negative thing that that gave you a clue that what we're dealing with a human hybrid, and it's very strange and odd. And it says here, you got from the DNA study, the documented anomalies came from DNA samples that yielded long sequences with pristine electropherograms at the other loci, including at least one AML-Y exon. Notably, this indicated that the DNA was of high quality and that degraded DNA was not responsible for the anomalies. Basically, they're saying it wasn't contaminated, and the DNA was not degraded or damaged or diseased or anything else. And so they had good pure DNA, but it was giving them really weird samples. And here's the chart here. I'll leave it up for a second. You can stop screen if you're someone who is clinically minded and would like to take a closer look at these different sequences. The nuclear DNA of the selected samples were tested for the following human genes. In other words, they went ahead and tested these for the MCR1R, which is a human Neanderthal red hair color gene. They also looked for the TAP1, which is the human antigen gene. They also looked at MYH16. This gene is associated with the sagittal crest in apes and was analyzed in an effort to determine if the unknown hominid was related to apes. So in the study, they were covering all their bases. So the results, the MCR1, which is the human Neanderthal red hair color gene, in the samples 28, 33, 35, and 37 had sufficient DNA extracted and were chosen for the MC1R sequencing. And the primers were used, the human primers were used for the MC1R gene. Were for apes, the primers used for the MC1R gene were apes, humans, and Neanderthals, because they had primers for each species. The samples were sequenced at DNA Diagnostics and Sasquatch Lab, two different labs, blind study. The DNA Diagnostics, the samples indicated a normal human MC1R sequence and carried alleles for the red hair color in humans. The samples were not from apes, from Neanderthals. In other words, they amplified using the human primers, but they did not amplify using apes or Neanderthals. That's from the DNA Diagnostic Lab. The Sasquatch Lab results partial human sequences in some of the DNA samples, while others had unknown sequences that were not human, ape, or Neanderthal, and still others failed to amplify. All human control DNA amplified and sequenced successfully from both labs. So Sasquatch got some really weird stuff of unknown sequences in there, and so they didn't amplify regardless of what primers were used. The YH16, this gene is associated with the sagittal crest in apes and was analyzed, like I said, for it to determine if it was an unknown hominid was related to apes. The primers used were both ape and human. The samples were sequenced. They did an ape and a human primers. The DNA Diagnostics Lab said all DNA samples that successfully amplified yielded results consistent with human and aligned with human reference sequences. In other words, they weren't apes. In the Sasquatch lab, all DNA samples were consistent with humans. So both of them, again, verified themselves. They were both humans and were consistent for humans. So it's not an ape. It doesn't have the sagittal crest gene. So the TAP1 human antigen gene is associated with breast cancer, diabetes, and several other syndromes. When they used the primers for this gene, some of the samples showed the TAP1 sequence and others failed to amplify. Unknown sequences were found in some amplifications with TAP1 primers yielded long sequence of unknown origin. Two sets of two samples had long unknown sequences that failed to match anything in GenBank, but interestingly matched each other. So let me read that again. Two sets of two samples had long unknown sequences that failed to match anything in the GenBank. In other words, they didn't match any other organism that exists. But when they were compared to each other, they matched. In other words, these two organisms, these two sets of samples that came from two different Sasquatch matched each other, which was a smoking gun to say. It's more than a smoking gun. It basically says these two individual Sasquatches are related, or they match. Not related as in blood relationship, but are the same species. Two of the samples, let's see, GenBank, but interesting match. These are two of these samples 33 and 44, matched with one another, although there were some SNPs that were inconsistent between the two sequences. These two samples were different in hair color, and obviously were from two different individuals, and were retrieved at different times. Samples 33 and 34 were collected from the same general area, and the donors thought to be related. Samples 10 and 43 also matched each other. Samples 33 and 44 TAP1 sequences were different than samples 10 and 43. Samples 33 and 44 were supposed to be related individuals, where samples 10 and 43 came out from different locations. So basically we got two sets. We got four samples, and two samples matched were related. It showed that we had individuals are actually related to each other. The other two just matched, but from different locations. In other words, the same species. I know that's a mouthful. We're getting down into the into the weeds, but I think it's important. And I know some people just eyes glass over and they jump out, and that's fine. But if you watch this, at least these terms are in your mind. What happened is in your mind. So when you encounter the disinformation campaign, which you're going to run into, you'll be armed with the truth and knowledge of what was going on and what really happened. So they also did a whole human genome SNP analysis, and so 24 test samples were tested for the whole human genome. That's 2.5 million SNPs. And they used the Illumina bead array 69 platform and the Illumina iScan instrument. All of the 24 samples failed to meet human thresholds of 95% SNP performance. The results range from 53 to 89. In other words, these genomes were not human. And they weren't animal. They were of an unknown species. Dr. Ketchum tried to duplicate these odd results by purposely degrading human blood. The samples were degraded with bacteria, and even though the samples were degraded, they still successfully tested at 97.15%, compared to the best that a Sasquatch sample would do was 89% of the non-degraded Sasquatch samples. Again, it's what's missing. You've got this very strange, you've got human mitochondrial DNA, you've got this nuclear DNA is showing human in some areas, showing unknown in other areas. So as they go through the different analysis, it's really all over the place. It's not human on the nuclear side. And it's not, where it should, if they took a human DNA and ran these tests, they'd all come back human. And so now, like I said, they've got a human hybrid going on here. That's the whole ideal here. That's basically what these results are telling you. To further rule out contamination and to address the possibility that normal human DNA samples could produce such markedly low SNP performance in the Illumina bead array assay, a separate group of human DNA controls was tested. These samples were extracted with buccal swabs obtained from sample handlers and laboratory workers, and were also subject to the identical bead array testing. These human controls came back at a performance above the 99th percentile level. In other words, just to make sure there wasn't something wrong with the equipment, they swabbed some lab workers and people that had found samples and ran them, and they all came back. In other words, what they're saying is if somehow it got contaminated, and therefore it ran at these very, very low levels, they were going to see if they could reproduce that from one of the handlers or somebody from the lab, and that's not what happened. In other words, everybody from the labs tested 100% human at 99%. So this very weird result of these low scores for these SNPs again was another glaring negative, is saying we've got a human hybrid that has very unusual DNA. And then here's the list of the different samples and the percentages as they came back. You see down here is the control samples, the male and the female. And this sample here was that was Dr. Ketchum's, and it was purposely degraded with bacteria. This is one that was not degraded. It was 96.63. Electron microscope examination of sample 26. So sample 26, which was the hair and flesh, was selected to be examined under the electron microscope. And the sample, was selected for its purity and lack of degradation. So the electron microscope characterization is considered superior to the biochemical techniques when it comes to an accurate topographical analysis of single and double-stranded DNA, such as contour length, width. In other words, they're looking at the DNA and they're able to actually, with the electron microscope, look at the DNA, the RNA, and look at the double helix. Now they sent these off to 2 different labs. to do the same analysis. So 2 labs got these, and the results came back for these long double-stranded DNA, 15 to 20 kb, but some of the DNA was also comprised of smaller fragments. Most of the DNA showed clustering of material along the length of the ends and were typical of disordered single-stranded segments. So you had DNA is double-stranded, and these samples that were pristine coming from the Sasquatch had disordered single-stranded segments. There was also frequent single-stranded gaps and single-stranded ends, which were not observed with the degraded human DNA control sample. In other words, they had a human control sample that they degraded on purpose, and even it didn't show these it didn't degrade to the point where it would go into the single-strand thing. Occasionally, with the single-stranded ends, a backfolding was observed that is typical for unspecific base pairing that could not occur in the absence of treatment with formaldehyde, glyoxyl, or elevated temperatures. Single-stranded DNA segments residing within double-stranded DNA. In other words, you had single-strand, double-strand, single-strand. To human science, to our knowledge, a living biological entity cannot exist with that kind of DNA. Yet, here we go. The Sasquatch does have this type of DNA. It's very weird and it's very unusual. The electron micrographs of the DNA revealed the unusual double-strand, single-strand, double-strand transitions, which may have contributed to the failure of it to amplify during the PCR. The high quantities of single-stranded DNA interspersed with double-stranded DNA may suggest substantial structural anomalies in the DNA itself. This is from the DNA study. This is their summary results. The DNA was visualized twice by electron microscope from aliquots, I can't pronounce that, of 2 different extractions from the same sample. Both of these extractions not only had yielded successful PCR applications previously, but were combined and utilized for the whole genome nuclear sequencing at a later time. Alternatively, the abundance of single-strand loci observed by the electron microscope could be evidence of genetic manipulation of the Sasquatch genome. In other words, all that fancy talk is saying this double-strand, single-strand basically it looks like the Sasquatch its DNA has been genetically modified, that it's been manipulated. So it's someone's cooked this thing up, or parts of it. And here's electron microscope. This is the actual double-strand, single-strand from the transitions from electron microscope. There's a graphic showing how you have the double-strand, you got the double helix going up, and then you have this branch where you get these single strands. It's very weird. Due to the whole sequencing here, the next generation whole genome sequencing, due to the large number of failures to amplify, failures to meet the human SNP threshold, and unusual sequences obtained, Dr. Ketchum selected 3 samples, 26, 31, and 140, with large amounts of high-quality DNA that were selected for the next generation whole genome sequencing machine. The DNA from these 3 samples was sequenced using the next generation Illumina platform at the University of Texas Southwestern in Dallas, Texas, a laboratory that sequences human genomes. In-depth analysis of all 3 genetic samples, that's 26, 31, and 140, was performed at the University of Texas Southwestern, and the alignment was confirmed by the University of North Texas Health Science Services. So you had 2 different places, you have 2 different university labs, looking at these sequences and confirming each other's results. This is a picture of the Illumina, the High Sequencer 2000. It's the machine that actually you put the samples in and it runs the DNA profiles. Q30 scores. Explain this because you may have some disinformation people out there try to say, they're going to say it was contaminated, and they're going to try to confuse the Q30 score issue. And basically the bottom line is the high sequencer 2000 gave scores. They call them Q30 scores. And the Q30 score will tell you the quality and the purity of the DNA sample. And basically, it actually gives, it's a digital readout. It gives you this number when you run the samples. And according to Illumina, this is their specifications. They say if you run a pure single source sample, should have a Q30 score of 80 or greater. In other words, if it's pure DNA, it's not contaminated, it's not mixed with someone else's DNA, but it's from a single source, it's not contaminated, then it's going to have a score of 80 or greater with an average of 85. However, if there was contamination, so basically the score, I can't read tonight. Basically, the score of 80 or greater means it's human. I mean, not human, means it's not contaminated. The Sasquatch samples had an average of 85. Actually had an average of 88. Sorry. However, if there was contamination, I just explained all that, blah, blah, blah. And if there was contamination, your score would be 40 to 50. The Q30 scores for the 3 Sasquatch genomes sequenced had Q30 scores of 88.6, 88.4, and 88.7. This is an electronic machine, uncontaminated, that did the analysis, and all the samples came back basically as pure single source. Period. In a sentence. They were single source, non-contaminated, and the quality and the sequence were far above the average. In other words, 80 is your average. So they were pure samples, and they were non-contaminated. So the results of this, all 3 genomes matched each other and were from the same unknown species. So they ran the 3 sequences, and these 3 sequences matched each other. They're related to each other. They're the same unknown species. The 3 genomes were compared to all the genomes in GenBank using the BLAST study, and they didn't match any known organism or any known anything in GenBank. So they're the same species. They match each other, but they don't match anything in GenBank. The sequences produced by the Illumina Next Generation platform match the mitochondrial DNA sequences extracted from the same samples by family tree DNA in the early stages of the study. In other words, they went back and compared the mitochondrial DNA from family tree DNA that had been extracted with what was extracted by the Illumina machine, and they matched. In other words, it's a way of double checking the work. And so, one lab, Family Tree, did it on using a different set of using primers and a different process, and then the Illumina process, which was the machine process, they basically have the same results, and so they verified each other. The genomes from samples of 26 and 140 were used to produce a primate family tree using the tool provided by GenBank called BLAST-N. This produces a graphic representation of where the genome falls in the primate family. And it does one of those trees, and it shows where this thing is. So you see here, like you have Homo sapiens on this tree, and it branches off. Well, over here on the Homo sapien branch is the Sasquatch samples, which come out unknown. And that's a statistically based family tree based off the genomes. So the DNA study summary. And this is as we say, we're going to boil her down here, and hopefully not put everybody to sleep. This is the most important thing. This is what you need to try to remember. As we, like I said, as the disinformation campaign and the personal attacks and all the crap starts, when the poo storm. Over a 5-year period, over 100 samples of hair, tissue, tree bark shavings, saliva, and blood were submitted by a nationwide team of submitters from 14 U.S. states and 2 Canadian provinces. All samples were screened prior to DNA analysis to eliminate common wildlife species using a variety of methodologies, including microscopic, morphological examination, and comparison against a large reference collection of North American wildlife hair. Over 100 hair samples passed the initial screen and were determined not to be human or from a known wildlife species. It was discovered during testing that the Sasquatch hair alone would not yield mitochondrial DNA. This was unusual because both human and animal hair will yield mitochondrial DNA. Only hair samples with ample skin tags were selected to be used in the study because that's the only ones that would amplify for mitochondrial DNA if they came from the Sasquatch. That tells you when you see these TV shows and they get a hair sample but it's just a hair sample, it doesn't have a skin tag, and it won't amplify, it comes back as unknown or won't run. That's the Sasquatch sample in the TV show. To avoid contamination, the hair samples were forensically processed and cleaned. Saliva, flesh, blood, tooth, fingernail, and enamel samples were also submitted to the project. The hair samples were sent to multiple labs for blind testing. All the samples were determined to be human, with the results consistent with human mitochondrial DNA. Non-human hair yielded human mitochondrial DNA. Said it 1,000 times. Results from purported Sasquatch samples tested over the previous 2 decades had always yielded a result of human mitochondrial DNA. These results were always dismissed as human contamination. The results of the Sasquatch DNA studies support the conclusion that these samples were in fact Sasquatch samples. 30 mitochondrial DNA samples were selected for complete genome sequencing and HV region haplotype sequencing results. In other words, we're going to find out where they're from, the regions of the world. The samples yielded haplogroups that showed the origination of the Sasquatches to be from Asia, Africa, and Europe. Only 4 out of the 30 haplogroups originated close to where the hair samples were contained in North America. Also, one of the samples contained none of the samples contained mitochondrial DNA from apes, Neanderthals, or Denisovans. The amyloidogenian gene from the nuclear DNA of the 30 samples was sequenced to determine the sex of the subject. Mixed results were obtained from human chromosomes, to others failing to amplify, while others would only amplify the Y chromosome. Again, pure, non-contaminated samples that yielded human mitochondrial DNA failed to amplify when human primers were used for sex determination. The samples were also tested for the MCR1 human Neanderthal red hair color gene and the MYH16 gene, which was associated with the sagittal crest in apes, and was analyzed in an effort to determine if the unknown hominid was related to apes, and the TAP1 human antigen gene associated with breast cancer, diabetes, and several other syndromes. The NCR1 result were human, no ape or Neanderthal sequences. The MYH16 results were human. The TAP1 results showed unknown when compared to the GenBank, but the samples matched each other. 24 samples were tested using the whole genome, that's 2.5 million SNPs, Illumina Beta Ray 69 platform using the Illumina iScan instrument. All of the 24 samples failed to meet the human threshold. Again, you can see this pattern where it will test human in one section of the gene and non-human in another. And that's basically what that's saying. A core sample of flesh was taken from sample 26. It revealed the unusual double-strand, single-strand we just talked about, and could suggest evidence that the Sasquatch had some sort of in the past had been genetically manipulated. The 3 samples contained ample nuclear DNA were selected for the whole genome sequencing. The DNA from these samples were sequenced in the next generation Illumina platform at the University of Texas Southwestern in Dallas, Texas. In-depth analysis for the University of Texas Southwestern, and alignment was confirmed by the University of North Texas Health Science Center. The result genomes were compared to the complete GenBank database using a blast search. No match was found. The 3 genomes were from a completely unknown species, yet the 3 samples aligned and matched each other. The conclusion, and y'all were going, phew, I know that's a lot. And that's dry. I know it's dry. But it's, sometimes important information's dry. And I'm trying my best to liven it up. So, I was going to have dancing bears on each shoulder, but I couldn't pull that off. So the DNA from over 100 separate samples of hair, tissue, toenail, bark scraping, saliva, and blood obtained from dozens of collection sites throughout North America have been extracted, analyzed, and sequenced. The hair samples were not consistent with human or any known wildlife hairs. Analysis of the DNA found that the mitochondrial DNA was human, but the nuclear DNA was of an unknown source. All known ape and relic hominin species such as Neanderthal and Denisovians were eliminated as being contributors to both the nuclear and the mitochondrial sequences. The whole genome analysis showed that the Sasquatch is an unknown species with some DNA sequences not matching humans or any known species, while other sequences did match humans and other primates. Here's the conclusion from the Sasquatch genome study itself. The data clearly supports that these hominids exist as a novel species of primate. Novel means unknown or undiscovered. The data further suggests that they are human hybrids originating from human females. This hybridization can be linked to humans with Denisovian admixture resulting from Denisovian males mating with human females. I don't know where I got the O3 from. The same type of mating potentially occurred with Sasquatch. Basically what it was is there was a study that showed that the human beings hybridized with Denisovians. And it's basically, she's saying that the same type of mating potential occurred with Sasquatch. In other words, just as the Denisovians and humans had a mixture resulting in males mating with human females. The same thing happened with the Sasquatch. You had whatever the originating male for the Sasquatch species mated with a human female. Though primary analysis supports the hybridization hypothesis, alternatively, it could also be hypothesized that the Sasquatch are human in origin, having been isolated in closed breeding populations for thousands of years. Nevertheless, the data conclusively proves that number one, the Sasquatch exist, and number two, that they are an extant hominid and are a direct maternal descendant of modern humans. So, I think that's all. Let me go over this real quick because a lot of people say, well, no one supports the study. Where are them scientists at? I see all these scientists say the study's bunk. Say it's BS. Every time I do a Google search, I have 45 pages of it's all garbage. Thank you, Google, for suppressing the information. But basically here's some actual, here's some support for the study. These are named people. They have they're doctors and they're experts in their field that looked at the study and are supporting the study. Dr. David Swenson, he's a biochemist, has over 39 journal publications to his credit. He looked at the paper and made the following statement. And he was actually evaluating Dr. Ketchum's work for Brian Forster and these other people. It says, Brian Forster, Jeff Cart, and other interested parties. I went over the manuscript by Melba Ketchum on Bigfoot genomics. My desktop had difficulty with the blast analysis of the consistent sequences. It helped me to understand more about the project. This collaborative venture has done a huge project that taxes me to fully grasp. I see interesting hominology with a standard human sequence with 99% match for mitochondria. From my abbreviated study, the nuclear genome seems to have human and non-human sequences. My opinion of the creature is that it is a hybrid of human mother and unknown human hominid male. Just as reported, for all practical purposes, it should be treated as a human and protected under the law. Brian selected the selection of Meldus Lab for your studies is a very good call. The Sasquatch is real, as proven by the genetic analysis. That was his statement. A study published by the journal Nature appears to also support the general findings of the DNA study. One of the main critics was the journal Nature, one of the main criticisms of the Sasquatch genome paper published in 2013 was hybridization between Sasquatch and modern humans was ludicrous or impossible or a wild speculation. But wait, fast forward to December of 2013 instead of January of 2013, and a group of scientists published a paper in the journal Nature that essentially claims that the genome of a Neanderthal skeleton they discovered was actually a hybrid and a mixture of modern human Denisovians, Neanderthals, and a potentially unknown hominid. So basically, they are saying the same thing. And so, from the journal Nature study, the new analysis finds that the genomes of the Hans, Chinese, and other mainland Asian populations, as well as Native Americans, contain about 0.02% Denisovian genes. The genome comparison also showed that Denisovians interbred with a mysterious unknown fourth group of early humans also living in Eurasia at the time. So we have this unknown human that's not in GenBank, they say, and so there was hybridization there. And this is the chart where they show the modern humans, Denisovians, and you can see the potential unknown hominid. The sources. World-renowned genetic scientist Dr. Tom Gilbert of the University of Copenhagen recently sequenced hairs from the purported orang pendek, a Sasquatch-like creature that lives in Southeast Asia. He received the hairs that were analyzed and determined to be non-human, or any known animal. When he tested the mitochondrial DNA, the hairs came back, the non-human hairs came back as human. Though critics claim contamination, Dr. Gilbert is a world-renowned professor of genetics, and I would assume he would know how to properly process DNA sample without contaminating it. Dr. Gilbert has over 125 peer-reviewed publications to his credit. He was recently lead author on a paper where the whole genome was sequenced from a 12,000-year-old Native American child. And so basically this Dr. Tom Gilbert found the same results that Dr. Ketchum found with the orang pendek. He had a non-human hair yield human mitochondrial DNA. And that's the end of the slideshow. So I hope this helps, and I can get said again, it's dry, but I think it's a necessary evil for those who are interested. And those who are not interested, they clicked and watched 30 seconds and blew on by, which is fine. This is not for everybody, but for those who are interested, who want to know what's going on and want to be armed with the truth, this is part 3 and the summation of the Sasquatch genome study. And like I said, soon, hopefully soon, there's going to be more DNA results released and more public access papers released. Not going to, Dr. Ketchum's not going to, publish them in a journal. She's just going to make them public access and say, okay, here are the results and here's what we found. And with all the relative and supporting evidence and labs and all the good stuff with the summation, so you can just read the summary and see what's going on. So that's what's coming. I appreciate your patience, and next time maybe I'll do something a little bit less. I think I'm going to jump back and finish up doing the series on the Sasquatch Awareness Project, and then just other things as things come up, and just deal with them. A lot of incoming emails and questions, and so going to continue to do that. So again, dry. And some of you are, I don't charge extra if you want to play this, to fall asleep at night. And I perfectly understand, and it's free of charge. I won't charge you any extra. for the insomnia relief. So with that being said, guys, y'all take care, and I will see you soon, and God bless.